To understand life, we have to start with the word itself. Biology comes from two Greek roots, bio, meaning life, and logos, meaning the study of it. But before we can actually study life, we run into a surprisingly difficult logical problem. We have to agree on exactly where the threshold of life begins and where inanimate matter ends. As humans, we rely on basic observation. We look at something moving or reacting and assume it is alive. In strict scientific terms, these visual instincts are flawed. This brings us to the concept of a biological characteristic. A characteristic is a trait we frequently observe in living organisms, like moving or getting bigger. It is a common occurrence, but it is not an absolute, unbreakable rule. That is distinctly different from a defining feature. A defining feature is a strict axiomatic rule. It must govern all biological life, completely without exception. In academic biology, and especially in competitive exams like NEET, you cannot rely on loose characteristics. You have to adhere strictly to defining features to prevent classification errors between what is living and what is dead matter. This 2x2 logic matrix represents the ultimate filter for biological traits. For a characteristic to be considered a true defining feature, it has to pass two strict conditions plotted on these axes. The first condition is universality, present in every living organism on Earth, no exceptions. The second condition is exclusivity, completely absent in all non-living matter. Traits hitting both marks land in this top right quadrant. Let's run our first test case, growth. Getting larger seems like a foolproof sign of life, but it fails the exclusivity test. Non-living things grow too. Mountains and crystals increase in mass through external accumulation. Because inanimate objects can technically increase in mass, we arrive at our first formal NCERT rule. Growth alone cannot define life. Let's run a second test case. Reproduction. Creating progeny seems definitive, but it fails the universality test. Many living organisms cannot reproduce. Sterile worker bees, mules, and infertile humans are alive, but do not multiply. This gives us our second strict rule. Reproduction is not a universal characteristic of life. Since visual traits like growing and multiplying have logical exceptions, surface-level observations are insufficient. We must look deeper into the invisible mechanics of the cell. This brings us to our third test case, metabolism. We are looking past external behaviors and focusing entirely on internal chemical reactions. Metabolic reactions are split into two types. First is anabolism, the process of building up complex molecules from simpler ones. Second is catabolism, the exact opposite, breaking down complex molecules to release energy. When tested for exclusivity, metabolism passes perfectly. No non-living object performs self-sustaining internal metabolic reactions. It also passes universality. From bacteria to whales, every living cell relies on this chemical engine. This provides the answer to one of the most critical exam questions you will face. Metabolism is the absolute defining feature of living organisms. Life, in scientific terms, is not defined by what an entity looks like. Rather, it is defined entirely by the continuous thermodynamic cycle of building and breaking matter within its cells. Once we have a strict rule for what constitutes a living organism, we face an entirely new problem: how to manage and organize the sheer volume of millions of discovered species. This flowchart illustrates taxonomy. Taxonomy is the scientific framework used to systematically organize the biological world across three specific stages. Stage 1 is identification. This requires observing an organism's traits to verify whether it is a previously known species or a completely new entity. Stage 2 is naming. Once identified, we apply a universally accepted scientific title to the organism, so researchers in any language can study it without confusion. Finally, Stage 3 is classification. We place the organism into specific structural categories based on its evolutionary relationships. The architect of this entire system was an 18th century Swedish botanist named Carolus Linnaeus. His work in structuring the natural world was so comprehensive that he is formally recognized today as the father of taxonomy. Linnaeus's most lasting innovation was a system called binomial nomenclature. Under this standardized convention, every scientific name requires exactly two components. It starts with a broader genus name, followed by a highly specific species identifier. If we apply this strict rule to human beings, we get our final high-yield data point. Our genus is Homo, and our species is sapiens, giving us the universal scientific name Homo sapiens. This gives us the complete two-step logic of biology. First, we use strict metabolic rules to determine if a piece of matter is alive. Second, we use Linnaean taxonomy to organize exactly where that life belongs in the natural world.